So you heard earlier from our keynote speaker that there's clearly an environmental component to longevity, and that due to all kinds of improvements in healthcare and interventions that over the years that longevity has increased and probably continues to increase. We also know that healthy lifestyle choices can extend lifespan by up to maybe a decade. However, to get to the 100, 110, 120, this is where we probably get into genetics and which versions of genes you inherit probably dictate that to some extent. We know, as Dr. Hodes just mentioned, there have been several initiatives to study genes and how they impact lifespan, particularly in model organisms. So Cynthia Kenyon started this out several years ago, and she found a very interesting thing, that if you mutated a single gene in C. elegans, the worms live twice as long. Okay, a single gene, they live twice as long. So if that works like that in humans, all we have to do is mutate one gene and we'll all live to be 160. It's probably not going to work quite like that, but that started the ball rolling on a very interesting series of experiments and investigations in yeast, flies, worms, and even mice that we think will probably translate into humans, but it's still a little bit early. So for example, the DAF pathway that I just mentioned that Cynthia Kenyon found has homologs in humans. And so that's been a very active area of investigation. And in particular, it's an insulin growth factor receptor pathway that is retained in humans. And so we think that's going to be a very promising thing. However, several lines of research converge. So for example, caloric restriction has also been observed to extend lifespan in model organisms. And so some of the pathways that people have uncovered in model organisms now, are good candidates for perhaps studying longevity in humans. So there are about 4 or 5 different pathways that people have identified now. And the common theme seems to be that these pathways sense cellular stress and they put the cells into sort of a protective mode. And that as a consequence of that, the lifespan is increased. So if it works like that in humans, we should be able to identify these same pathways and study them. And so that's where my lab got interested in this, and we started to study and sequence the genomes of people who live to be at least 100 years old, called centenarians. And there are a number of groups doing this now. Some humans also live, as you heard from the earlier stories, to be super centenarians, or they live beyond 110, and maybe even up to 122 or beyond. And that's a big question. How far can we live? Chimps have an average lifespan of about 30 years in captivity and probably less out in the wild. And so if you think about a common ancestor 6 million years ago, chimps are at 30, we're at about 80 now. So something happened during that time, and probably part of it is, health and civilization, but other things, part of that could be genetics. So as we move forward studying the centenarians, we've now sequenced the full genomes of 60 centenarians, trying to understand whether specific pathways are mutated or whether people might inherit specific alleles of some of these same genes in these pathways. And I think to some extent the answer is going to be yes, and to some extent the answer is going to be it's probably a little bit more complicated in humans because we have more copies or more versions of genes than roundworms. We've often duplicated genes 2 or 3, 4 times. So these pathways have diverged. One area that there's a clear overlap, and you'll hear from Dr. Greider, is in telomerase. But some of these, the question will become, how many ways can you become a centenarian? And if it's 2 or 3 ways, then it will be pretty easy to figure out. But if it's like a lot of human phenotypes, it may be more difficult, and we may need to sequence hundreds or even thousands of these centenarians to really get a good answer. So back to centenarians and why they're interesting. We're sort of coming at it from the opposite end of the spectrum in terms of, we're saying they have this long lifespan. What genes influence that? And I think there are a lot of implications underlying that question. So for example, health span is something that people talk about that may or may not be linked to lifespan, where a lot of the ways these centenarians seem to live to these extended ages is by avoiding common diseases that account for most mortality. So for example, centenarians avoid cancer. Cancer incidence alone is higher in non-centenarians, but also if centenarians get cancer, they tend to survive it. And they don't have such advanced stage. They typically don't get metastases, for example. So how do they resist cancer? I think that's a very interesting question. They also, as you saw from the keynote address, that people with Alzheimer's disease tend to have, a shorter lifespan, or the flip side of that is centenarians likely evade getting Alzheimer's disease. So maybe these centenarians hold a lot of secrets about how to live a long, healthy life, that if we can understand them, perhaps we can mimic them in regular people with targeted therapies. For example, if we could take some kind of a compound every day and live to be 100 and never get any of these major diseases, that might be a great thing and save the NIH a lot of money. Well, but seriously, there's this idea of compressed mortality where you would live a much longer healthy lifespan, and then these mortality issues would come up, but in a relatively compressed period of time. So other questions about centenarians, do they, the maximum lifespan, can we live to be 150? Can we push it to 200? That's another question maybe we can talk about as we open it up for discussion. And I think I'll pass it off to Dr. Greider at that point.